Hello, everyone. Thank you for joining us today. My name is Dr. Tim Abbott, and I am IACCE's Associate Director for Youth and Public Outreach, and am a postdoctoral fellow at the University of Waterloo in Ontario, Canada, and I will be moderating the seminar today. I would like to extend greetings on behalf of IACCE's President, Dr. Hung-So Lee, and the entire IACCE executive team and staff. Before we begin, I'd like to take a moment to talk a bit about IACCE and to provide some background. IACCE was established to promote the development of a circular, science-driven economy. A circular economy entails gradually decoupling economic activity from the consumption of finite resources and by designing waste out of the system. This process mimics nature, in which waste from one process is the feedstock for another. A circular economy is regenerative by design and reimagines the traditional extract, consume, and dispose economic model, and instead redefines growth by focusing on positive, society-wide benefits. Now, IACCE was established to promote the development of advanced knowledge, technologies, and policies in partnership with industry, academia, and governments, and to provide a platform for the exchange of knowledge. And developing a circular economy will require multiple key and intertwined areas and industries upon which IACCE is primarily focused. And we invite everyone in attendance to consider becoming members. Individual memberships are open to all, and we currently have members ranging from students to professors, engineers to government officials, and business executives from a wide range of industries from over a dozen countries around the world. Membership is currently free and offers a wide range of benefits. Please visit our website for more information. And now I'm delighted to introduce tonight's keynote speaker, Dr. Michael Dorrington. Mike has been a Strategic Partnerships and Resource Development Advisor with Ontario Genomics since 2020. He supports the implementation of strategic initiatives to bring new genomic-derived solutions to key sectors of Ontario and beyond. Mike has a PhD in Infection and Immunity from McMaster University in Ontario, Canada, and a Bachelor of Science in Life Sciences from the University of British Columbia in British Columbia, Canada. Before joining Ontario Genomics, Mike was a postdoctoral fellow at the Laboratory of Immune System Biology at NIH's National Institute of Allergy and Infectious Diseases near Washington, D.C. Here he used cutting-edge genomic and transcriptomic solution technologies to study antiviral cytokines in inflammation and antibacterial immune responses. On behalf of IACCE and IACCE student chapters, welcome, Dr. Dorrington.
Thanks so much, Tim, and a big thank you to both Tim and Young-Sul for the invitation to speak with you all today. So as Tim mentioned, I'm Mike Dorrington, and I'm an advisor at Ontario Genomics, and I'll be talking to you today about the exciting field of engineering biology, which you may have also heard referred to as synthetic biology. So my goal today is to give you a little bit of an overview of what has led to this exciting time in the engineering biology space, as well as our vision for Canada's place in the future bioeconomy. And before I get into that, however, I want to take just a moment to talk about the Canadian Genomics enterprise, of which Ontario Genomics is a part. Okay, so the Canadian Genomics enterprise, or the G7 as we call it, is made up of our national parent organization, Genome Canada, and 6 regional genome centers, including Genome BC, Genome Alberta, Genome Prairie, Ontario Genomics, Genome Quebec, and Genome Atlantic. These 7 nonprofit organizations work together to ensure that Canada maintains a world-class genomics ecosystem through research, innovation, and what we call stakeholder management. So these stakeholders for us include the government, research institutions, companies, schools, and the public at large. We serve these stakeholders through many mechanisms, including the funding of research projects, the building of coalitions, such as the Canadian COVID Genomics Network, or CANCOGEN, by acting as expert consultants to federal and provincial governments, as well as supporting industry, especially through investments in small to medium enterprises and startup companies. We also provide services to improve genomics-related education and outreach initiatives, for example, by partnering with educational organizations on lesson plans and informational videos related to genomics. Finally, we do all of this while always maintaining a view of how our work impacts our broader society by always thinking about what we call GELS. So this stands for genomics and its ethical, environmental, economic, legal, and social aspects. Now, the G7 are involved in the application of genomics to any industry or sector that has anything to do with living organisms. This includes health, agriculture, the environment, natural resources, and energy. And I'll be talking about a number of those sectors today. We do all of these things at Ontario Genomics, but with a particular focus on our region of Ontario. We help Ontario researchers and companies get funding for transformative genomics-based or genomics-enabled projects, work with the provincial government to build world-class research and innovation consortia, and build lasting education and outreach programs across the province, all in the name of supporting our vision of healthy people, a healthy economy, and a healthy planet through genomics innovations. Our end goal is to provide leadership across sectors that are important to Ontario and Ontarians, and especially health, agriculture, natural resources, and manufacturing. And we do this primarily through the development of public-private partnerships. That is, partnerships between academic and industry leaders, using the talents and resources of each to solve some of society's most pressing challenges. Now, our work has supported the injection of over $2.5 billion into Ontario's genomics ecosystem through the funding of 230 plus research projects involving well over 500 partnerships. Now, one of our most recent contributions to date is the formation of the Canadian DNA Engineering Systems Network, or CanDesign, which is a group of over 80 organizations that is led by the National Engineering Biology Steering Committee. Chaired by our CEO, Bettina Hamlin, the steering committee are working to drive a transdisciplinary roadmap for engineering biology in Canada. The ultimate goal of this committee is to ensure that Canada takes a leading role in what we call the biorevolution. So what do I mean by biorevolution? Well, the biorevolution is really just the replacing of industrial processes, many of which rely on the consumption of fossil fuels, with biological systems. So the underlying technology in the biorevolution is what we call engineering or synthetic biology. And this is at the core, which at its core, is the application of engineering principles to designing biological systems. So the McKinsey Global Institute estimates that up to 60% of the world's physical outputs could be made using biological systems, with microbes like bacteria and fungi, as well as plants, being the most common organisms used for this purpose. Some examples of applications of engineering biology include producing meat without using any actual animals, the production of important pharmaceutical compounds and vaccines in plants, and the production of bioplastics without the need for fossil fuels as the starting material. Now, this new bioeconomy is estimated to provide up to $2 to $4 trillion per year in economic value within the next 10 years, giving it the potential to completely change how we provide the world with its needs. So what's driving the relatively sudden increase in support for engineering biology as a platform? Well, the keys here are our abilities to both read and write DNA in an accurate and cost-effective manner. One of the key contributors to this is the absolute explosion of genomic information now available to us, which is being driven by ever-decreasing costs to sequence a genome. The Human Genome Project, wherein the majority of the human genome was mapped for the first time, took about 13 years and $2.7 billion to complete. In the 20 years since then, novel technologies such as next-generation sequencing have allowed us to sequence the entirety of an organism's genome in a matter of hours and for less than $1,000. Concurrently, other technologies are unlocking more than just our ability to read the genetic code, but actually understand its full potential. Now, the central dogma of life dictates that genes are transcribed into messenger RNA, which are then translated into protein sequences. A relatively small number of genes can be combined, spliced, and rearranged to create a number of proteins more than an order of magnitude greater than those genes themselves. For example, humans have between 20 and 25,000 coding genes, which give rise to over 1 million different protein structures, each with a specific role within the cells of our bodies. Genomics, transcriptomics, proteomics, metabolomics, and other big data biological disciplines are key to understanding how biological processes are created, combined, and governed. We can't build systems that we don't intimately understand, so we need to know how changing the genome of an organism will affect these other molecules of life. How will changing the expression of a single gene affect the rest of the genome? How will this affect the transcriptome and proteome of that organism? Will this lead to unintended consequences? These are all important questions as we begin to create biological systems for real-world purposes. Now, one thing that I like to say is there aren't many problems that nature hasn't already been forced to solve. Life has endured these past billion years due to constant evolution, allowing for increasing levels of complexity, driven by semi-random mutation of DNA, coupled with the force of natural selection. The enormous variety of life that has emerged through these processes now provides us with nearly limitless potential to mine the genomes of organisms, to engineer novel systems from collections of disparate genes. Take this metabolic map of a human cell, for example. Thousands of chemicals and proteins are constantly interacting in order to keep a cell alive and performing its tasks efficiently. By combining the metabolic systems of multiple organisms, editing how particular enzymes are produced, and/or deleting entire metabolic pathways, we can now fundamentally change the inner workings of an individual cell. As we learn more and more about the complexity of biological systems that occur in nature, we are now gaining the ability to harness the power of this complexity for our own needs. Understanding how the relatively simple sequences of the genetic code can govern the structure and function of cells, organisms, and indeed entire ecosystems is one thing. The ability to accurately and efficiently edit and write that code for a particular output is another challenge entirely. Novel technologies like CRISPR-Cas9-based DNA editing are now allowing scientists to edit DNA with increasing fidelity. DNA bases can be added, deleted, or mutated with relative ease. CRISPR-based screening approaches are also allowing us to perform millions of experiments in millions of cells at a time, further increasing the complexity of the biology that we're able to analyze. We're also gaining new capabilities for synthesizing DNA sequences from scratch. New synthesis technologies, countless omics datasets, and machine learning algorithms are coalescing with engineering principles to allow us to truly engineer biological organisms. By harnessing all of this biological complexity, we can guide living organisms to produce essentially anything we need. A microbe, like a bacteria, could be engineered to take in a carbon-based input, like a sugar, and through natural biological processes, create a cornucopia of outputs. This is already being done for a number of applications. In health, these technologies are key to producing the spike protein encoding mRNA in our COVID-19 vaccines. Alternative protein companies like Impossible Foods are using engineering biology to produce the iron-binding protein heme, which gives their ground meat alternatives the bloody taste and texture that consumers are accustomed to. Clothing companies are skipping the need for silkworms when they produce engineered silk fibers from bacterial chassis for use in clothing. I'll delve a little bit deeper into other examples in a moment, but these are just three of the countless ways engineering biology can replace what are traditionally climate-affecting, fossil fuel-consuming, and/or unscalable processes that have been in use for hundreds to thousands of years in some cases. Now again, there isn't much nature hasn't already been forced to solve. There are bacteria capable of breaking down plastics more efficiently than any thermochemical process we've come up with. Our immune systems are capable of responding to nearly any foreign entity that may enter our bodies. Yeasts convert sugar into alcohol and carbon dioxide with ease. Now, harnessing and augmenting these abilities creates a platform of innovation with nearly boundless potential. Humans have used biomanufacturing since the beginning of civilization, taking advantage of natural bioprocesses like alcoholic fermentation. We later industrialized these processes that organisms like yeast do naturally to make biofuels. Now, using the latest developments in biotechnology, we're able to take this approach a step further and engineer nature to produce the medicines, foods, and fuels that underpin our society, while addressing many of our current and future needs. Needs like becoming better stewards of our planet by not only protecting, but supporting and enriching the environment. Needs like making high-quality, cutting-edge healthcare more accessible and powerful. And needs like our need to feed 11 billion people by the year 2100, providing quantity and quality nutrition to the masses. And we need to do all of this while supporting and sustaining the global economy. To do all of this, we need to rely on some core principles of engineering biology. The first is modularity. So when building new bioprocesses, we don't always need to start from scratch. By building biological systems with mix and match parts, we can adjust for different inputs and outputs. The next is tunability. So it's important that all engineering processes are tunable, and engineering biology is no exception. By integrating molecular switches, gates, and amplifiers, this will allow for tunability of metabolic processes depending on the environment. Sustainability. So a core focus of engineering biology is reducing our impact on the environment through limiting greenhouse gas emissions, weaning us off of fossil fuels, and creating products that don't cause damage or become waste. Many engineering biology processes are centered on establishing the circular economy that Tim was just talking about, wherein waste from one process or output becomes the input for another. And finally, engineering biology needs to be decentralized. So this is both theoretical and physical. Engineering biology processes should be sector agnostic and able to run anywhere in the world. We've seen through the COVID-19 pandemic that our society is supported by increasingly complex supply chains, and engineering biology can support supply chain robustness by allowing us to quickly pivot among countless products based on need. So nature has provided us with the means to engineer a near infinite set of tools to support our planet and our society. But do we have the ability to capitalize? To do so, we need to change the way we think about science. Engineering biology is multidisciplinary or even transdisciplinary. It's also cross-sectoral and iterative, meaning we need people with strong backgrounds in a number of disciplines to apply their skills to this new platform. We need transdisciplinary thinkers leading multidisciplinary teams, and we need to bring together different market sectors to solve common problems. So let's take cellular agriculture as an example. So who knows the most about growing cells and tissues in a lab? Well, it's probably not current food manufacturers, and in fact, it's more likely to be the biomedical research community who does this on a daily basis. So we need to be able to leverage those skills. Okay, so now I want to delve a little bit into some specific applications for engineering biology across a number of traditional economic sectors, and I'm going to start with health. So I know that everyone here is sick of hearing about the pandemic, and I am too. It's altered our lives irreversibly and will have lasting effects on our collective health. However, the pandemic has also highlighted so much of what is needed to create a more resilient society, and how engineering biology can rise to meet these challenges. Genomics, artificial intelligence, and engineering biology were at the forefront of vaccine development against SARS-CoV-2, as well as the backbone of diagnostic testing and surveillance, especially through the sequencing of wastewater. Supply chain disruptions have meant empty store shelves, long delays in procuring scientific supplies and equipment, and a lack of appropriate personal protective equipment, both for healthcare professionals and society at large. Our reliance on single-use plastics has been exacerbated, accelerating an already out-of-control accumulation of these materials in our landfills and environments. None of this is COVID-19 specific, of course, and we must be better prepared for the next threat to our way of life. Whether this is climate-related, another biothreat like antimicrobial resistance, or anything else, engineering biology as a platform technology can help fortify supply chains, allow us to mitigate damage, and support the economy at the same time. Now, genomics and engineering biology aren't just applicable to viral pandemics, however. They're also key to realizing the dream of the original Human Genome Project. At its beginning, it was believed that once we could read the entirety of the human genome, we'd essentially be able to cure any disease with a genetic element. Now, this has proved more difficult than anticipated, but through the expansion of this vision past genomics, we're getting closer to this goal. Whole genome sequencing of patients and genome-wide association studies provide clues as to the genetic causes of some difficult to diagnose diseases. And engineering biology platforms are allowing us to target mutations and in some cases even fix them. One example is the CRISPR-based editing of patients with sickle cell disease. Altering one copy of the sickle cell gene in the stem cells of patients, scientists have found a way to potentially eliminate this disease entirely. In the cancer arena, we are now able to remove immune cells from a patient's body, re-engineer them to specifically target their cancer, then re-administer them. And sequencing technologies are also allowing us to track the rise of antimicrobial resistance by targeted sequencing in places like hospitals and long-term care homes, as well as a more bird's-eye view by monitoring wastewater. The hope is that while we can never fully know where the next threat will come from, at least we can stay better informed in the future and plan accordingly. Now, moving on to agriculture. So agriculture today is at a bit of a tipping point as we enter the fourth agricultural revolution, wherein technology will play an outsized role in how we feed the world. Ecological and animal welfare concerns about the way farming is done, climate change and shifting consumer preferences, and the need to feed 11 billion people on less and less land are all contributing to this. So in order to address these concerns, novel food production methods are needed, as well as more incremental changes to current agricultural practices. And this brings me to cellular agriculture. So cell ag, as we often call it, covers both precision fermentation as well as tissue engineering and cell culture. In precision fermentation, cells, like we've been talking about, are turned into miniature biological factories and used to produce a desired product in brewery-style tanks. This could be a more complete product like egg white protein or honey, or simpler ingredients like flavorings or pigments. In tissue engineering and cell culture, cells from animals, fish, or other seafood are grown outside the body to produce cultivated meat and seafood products, essentially creating meat without the animal. So here are just some examples of products that are in development and in some cases on the market around the world that can be produced through fermentation or cultivation. Fermentation-based rennet in cheese making has been produced since the 1990s. Dairy protein in products such as ice cream and cream cheese, as well as that heme protein in the Impossible Burger that I mentioned earlier, as well as plant-based products such as chocolate and coffee can all be produced through fermentation. Now, many of these products are already quite common. You may have tried an Impossible Burger, which is widely available here in Canada, and you might have heard of Perfect Day, who make ice cream and cream cheese so far. Now, cultivated meats are a bit further behind, but catching up quickly. In fact, hybrid chicken bites made of cultivated chicken and plant-based ingredients are now available in Singapore, while people are working on other products such as beef, fish, and foie gras. So why are we so excited about this space? Well, the types of products that can be produced is essentially limitless, and over $9.7 billion has been invested in this space in the last several years. And there are estimates that alternative proteins will reach a market of $290 billion by the year 2035. So we want to help ensure that Canada is an exporter of these products and not just an importer of what others are already making. I also want to highlight that these are all meant to supplement more traditional agriculture, not replace it entirely. To feed those 11 billion people, we'll take all of the tools we have along with others that we've yet to invent. Now, there's a huge issue in the agriculture industry, and that is food waste and other agricultural waste. So food waste accounts for up to 50% of the methane production associated with the agriculture and food sectors, most of which comes from it decomposing in landfills. Now, there are many ways in which engineering biology can help mitigate this issue, including engineering crops that have longer shelf lives and therefore are less likely to be thrown out before they're consumed, producing more food ingredients through precision fermentation, therefore decreasing valueless byproducts, or else engineering biological systems capable of using this waste as an input for bioconversion to value-added products. These products could include any number of carbon-based structures, including biodegradable plastics, textiles with minimal carbon footprints, biofuels, and chemicals like difunctional acids. I just mentioned bioplastics, and these are a very important class of materials that has the potential to both wean us off of fossil fuel-based plastics, but also limiting the amount of plastic that ends up in our landfills and environment. You've all likely heard recently that only 9% of all plastics that reach the end of their life end up being recycled, with 86% of plastic ending up in landfills. This is not a new issue, and our world is consistently being polluted with more and more plastic each day. So what can engineering biology do to not just slow the buildup of these materials, also deal with the plastic that we already have? Well, the answer may once again lie in the abilities of microbes. So there are particular insect larvae that are capable of subsisting entirely off of the carbon that they can leach from plastic. In the lower right of this slide, we see the larvae of the greater wax moth, which is one example of these plastivores. These insects are able to consume plastic due to the bacteria that live in their guts. However, these bacteria break down plastic at very slow rates, and so couldn't be directly applied to our recycling infrastructure at any meaningful scale. However, by engineering these bacterial communities to boost their plastic-reducing powers, researchers are attempting to make this process significantly more efficient, potentially giving us, in conjunction with fully biodegradable plastics, a pathway to zero plastic waste in the near future. Currently, textile production is also a huge factor in our battle for the environment, contributing up to 10% of the world's greenhouse gas emissions annually. And much like plastics, these textiles almost all end up in the landfill at the end of their life. Nylon generated from oil is an especially big opportunity here due to the high levels of nitrous oxide that its production releases into the atmosphere. Now, nitrous oxide is up to 300 times more damaging to the ozone layer than carbon dioxide. Reducing our reliance on fossil fuels, limiting greenhouse gas emissions, and dealing with the world's waste crisis will all contribute to our improved stewardship of our planet. Doing all this while creating well-paying, meaningful jobs is a challenge, but one that engineering biology is poised to address. For example, by replacing the need for fossil fuel inputs and reducing the number of chemical processes needed to reach the final product, the production of nylon from inputs like sugars by engineered yeasts can reduce greenhouse gas emissions by up to 70%. And these materials are not necessarily bound by the parameters of their natural production. For example, spider silk has many properties, including its tensile strength, elasticity, and others, that would make for great textiles while also being fully biodegradable. However, the proteins that make up spider silk are nearly impossible to weave into fibers, as was discovered when scientists attempted to express these proteins in the milk of goats. However, the tunability and modularity of engineering biology can allow us to alter the basic structure of the silk proteins to make them easier to spin into fibers. We can then express these proteins in any number of organisms, including plants, to be farmed. So most of the examples I've given so far involve the manipulation of microorganism-based chassis. These are workhorse strains of lab bacteria and yeast that we know enough about to be confident in perturbing their genomes. However, theoretically, any living organism can be altered through the technologies being developed today, including plants. Molecular farming, which is already taking place for producing certain pharmaceuticals and food ingredients, is an even more efficient way of producing many proteins and other molecules. In molecular farming, a gene of interest is inserted into the genome of a plant, which is then grown as normal. Upon harvest, chemical processes are used to purify the product of interest, leaving behind the rest of the plant material for other uses. Now, a secondary benefit of molecular farming is that the plant host can be chosen strategically. This could provide farmers with robust crops that can be grown on relatively inhospitable land, opening up opportunities where we cannot grow food crops. Finally, engineering biology has applications in information technology as well. So by the year 2025, humanity will have produced about 33 zettabytes of data. So that's 3.3 times 10 to the exponent 22 bytes of data. The infrastructure necessary to contain that much binary code is astronomical and requires an unsustainable amount of electricity and other factors. But by simply switching from a binary code to a quaternary information storage, such as the A's, C's, T's, and G's of a DNA molecule, 33 zettabytes could theoretically be contained in something the dimensions of a ping pong ball. In fact, all of Facebook could be contained in a poppy seed. Now, there are reasons why this isn't our current reality. DNA-based information storage has much higher error rates than traditional data storage, and there is significantly increased complexity in the reading and writing of that data. However, these shortcomings are the focus of intense research, and new technologies are being created to make DNA data storage commonplace. Okay, so I want to end off here by looking to the future a little bit. Engineering biology, like all cutting-edge technologies, hasn't had all of its kinks worked out as of yet. There are both scientific and non-scientific roadblocks to ensuring its widespread adoption. Scientifically, probably the biggest issues surround the idea of scale-up. As you can imagine, a group of microorganisms will behave differently in a 1-liter flask than they will in a 10,000-liter bioreactor, for example. Microclimates, nutrient accessibility, and gradients in temperature and pH all have profound effects on the ability of these organisms to perform at their best. Fermentation, the core process by which these processes work, remains quite water and energy intensive, meaning there is a ways to go before engineering biology can be truly resource neutral. And there's a great deal of optimization of metabolic pathways, culture media, bioreactor parameters, and more to be done for each input to output system we are creating. On the non-scientific side, engineering biology still lags behind other technologies as far as private and public investment. There's also a severe lack of infrastructure with which to experiment with and pilot new biological systems. There are also questions as to how to properly regulate the products of engineering biology, as well as questions of public perception and what Canada's national vision entails for these new technologies. Now, despite these roadblocks, engineering biology is already big business and has been for the past 5+ years. Even in 2017, and only looking at the US, biotechnology driven by engineering biology accounted for over $250 billion in revenues. So that means that $2 to $4 trillion figure I gave earlier doesn't seem so crazy when you think we were at almost a quarter of that 5 years ago. This is being reflected in real time even in Canada when looking at recent venture capital investments in engineering biology companies. The energy company Suncor has made significant investments in LanzaJet, who produce biofuels for the aviation industry. The Canada Pension Plan has invested in the animal-free dairy company Perfect Day that I mentioned earlier. And Toronto-based Arcturn Ventures led the Series B funding of Mosa Meats, among many others. But is private investment leading to public investment? So other technologies like artificial intelligence and quantum computing have seen significant federal investment as well as formal strategic planning, while engineering biology has lagged behind due to a lack of a national plan of action. We've certainly seen an increase in public investment in engineering biology in other jurisdictions, and in each case it has all started with a roadmap. So the US-based Engineering Biology Research Consortium, or EBRC, roadmap led to $87 million of federal investment in the BioMade Consortium, one of 15 nationalized manufacturing institutes in America. At an even larger scale, a synthetic biology roadmap from the UK led to government investment that has created more than $1.5 billion in startup investments, which is still growing. But perhaps the best comparison for Canada in this area is Australia, who recently released their roadmap for synthetic biology. Australia's National Science Agency estimates that by 2040, engineering biology could bring in excess of $27 billion of annual revenue and support up to 44,000 jobs. Interestingly, they believe the top opportunity lies in food and agriculture, including cellular agriculture. This mirrors the McKinsey report I talked about earlier, which showed that food and agriculture show the most potential for growth due to engineering biology. So despite the fact that health and manufacturing may dominate the headlines in this area, there are massive opportunities across sectors for engineering biology to have an impact. Canada has everything we need for a strong engineering biology development. We have the natural resources, and maybe more importantly, the biomass that we can use to convert into valuable products. We also have research expertise across applicable fields, strong industry know-how and need, as well as strong international trade relationships. So the question really becomes, do we have the will to follow up with this? So some key takeaways here. So engineering biology is the application of engineering principles to designing biological systems. It has worldwide applications across industries, including health, agriculture, manufacturing, and information technology. Engineering biology gives us the potential to wean us off of fossil fuels, reduce greenhouse gas emissions, improve animal welfare, and remove waste from the environment, among many other things. However, there's a need for a pan-Canadian engineering biology strategy to keep pace with the rest of the world and transdisciplinary training to create the highly qualified personnel that we'll need. Now, just finishing up, I'd like to point you to two resources that we've created over the past 18 months. So the first is a white paper describing engineering biology and Canada's post-pandemic opportunity. And the second, which was released just this past November, is an in-depth report on the state of cellular agriculture in Canada, along with a strong economic analysis of its potential benefits to the Canadian economy. If you'd like to dive a little deeper into anything I've said today, these are great resources to start, and both can be found on the CanDesign website. So with that, I'd like to thank you again for your attention, and again to Tim and Young Sool for the invitation. And I'd be happy to answer any questions that anyone might have, and I will stop sharing my screen.
All right, thank you very much for the extremely interesting and great presentation there, Mike. All right,
Thanks.
so I think we can open it up to some questions. So if you would just like to put your hand up or put a question in chat, I can bring you up in order. I see first question from Peter. Go ahead.
Hi, Mike. Thanks very much for the presentation. It's always very interesting to see what all the different possibilities are and the different things that are being pursued.
Yeah, for sure.
I guess having a little bit of experience with bioplastics and some other bioproducts, the issue we always come up against is like what feedstocks to use and how energy intensive they are to create in the first place for feeding with raw sugars. So I guess, what is the strategy generally for food production if we need to produce food to feed our bacteria in the first place?
For sure, yeah. So it's a great question, and yeah, that's we tend to focus on the core technologies and not necessarily what's feeding into them from the beginning or what's going to happen after, right? But there's still the need to ship things around, for example, which is obviously very energy intensive. So I think this really gets to the core of what we're trying to do with this circular economy, right? And we're really trying to find ways to convert biomass that already exists, so things like food waste, things like byproducts from the forestry sector, from pulp and paper mills, for example, and these really carbon intensive but not necessarily easy to kind of handle materials, right? And so that's a really key thing. Is you're right, the feedstock is just as important, and so we need to be developing technologies, and certainly people are, for taking that waste, collecting it all into one place, and turning it into that feedstock for the microorganism, right? So yeah, that's a $1 trillion question right there, Peter, and I hope you figure it out. But that's kind of what everything that we're trying to do is based on, that idea of taking waste from these other processes that aren't going to stop anytime soon, right? And then kind of creating the food for these microorganisms. And I will say, this is going to be a phased process, right? This isn't going to be all of a sudden we can go completely become a carbon sink, in the next 2 years or something. But a slow kind of progression. I think that we'll be able to get there in time.
And is there like a strategic framework for that adoption that's addressed in the white papers, or?
Yeah, so we talk about that a little bit in the white paper, but the short answer is that there isn't a very strong kind of strategy around that, as far as we've been able to tell. A lot of the focus has been on those core, building the microbes that will be able to take things in and give us the outputs we need, right? Without as much investment in kind of the ancillary processes that can drive engineering biology, and that's a really key question. And without those, then this isn't going to work, right? So there needs to be more public investment, I think, into these kind of the opposite ends of the pipeline, and not just the pipeline itself.
Thank you very much.
Thank you, Peter.
Thanks, Peter. All right. If anyone else has a question, please feel free to put your hand up on Zoom. One question actually I might ask while I'm at it is, what would you see in terms of challenges in terms of IP? So what does the IP landscape look like?
Yeah, IP is a mess no matter what industry you're in. I think that's fair to say. This is one of those. It's almost a little bit of the Wild West, right? Of everybody's scrambling to generate IP and register it and protect it in multiple ways. What I would love to see is a little bit more openness and sharing, like we've seen in other things, like for example, the Structural Genomics Consortium is a good example where we're all protein sequences are being shared widely and openly among researchers and things like that. But anytime there's money involved, and big money at that, there's going to be IP concerns, and I don't quite think that really we have a handle. Like I was saying with regulation and things like that, right? We don't quite have a handle on how to deal with all of this. And so again, those social sciences and looking at things like regulation is just as important as the technical know-how that's going to go into creating these processes. And again, something that people don't necessarily want to invest the time and money into, and governments in general tend to be slow on the uptake for these things. So someone's going to figure it out, and hopefully it's it's kind of the right people who want to put this technology in as many hands as possible and make it as open and accessible as possible.
And is there any jurisdiction that is doing maybe a better job than most in terms of that, or is it still kind of a problem kind of everywhere?
Yeah, it's a good question, and one that I don't have a really good handle on as of yet. But these things are just so new, right? I think with kind of sub technologies like cellular agriculture, certain jurisdictions, I think Israel especially is really kind of at the forefront of both that technical and the kind of these other things that we're talking about. But yeah, outside of that, I don't have a strong answer for that.
Okay. Right. And Vikas, I think I saw you wave your hand earlier. Did you have a question?
Yes, Tim, I have a question. Thank you. Thank you, Dr. Mike, for a very wonderful presentation. I have a very general question. I mean, when it comes to genetic modification, people always have a question of genetically modified, foods, which people feel like it might create some trouble in their body when we keep taking it. So my question is, like, what I believe, well, like what I have a very little knowledge when it comes to genetic engineering. Basically, it's mostly enzyme, like whatever is been done is like enzyme related technology, like all genes produces enzyme. So my question is it not possible, just give your highlights on that? What do you think about it when it comes to genetically modified food? Is it not possible that we can take out some, like we can just work on enzymes instead of just working on our living organisms? So maybe is there any type of technology where people just working on enzymes, taking that's just enzyme, not the whole microorganisms, and then that food will all still considered as a genetically modified food or not? So that's my question. Thank you.
Yeah, so it's interesting, right? I think so. There's almost two questions there, and one's easy to answer, and that is the whole idea of kind of cell-free processes, right? That are just based on the addition of successive addition of particular enzymes, things like that. That's absolutely in use. It's absolutely being worked on. Quite complex kind of combinations of enzymes can be used, but nothing quite recapitulates the environment of a cell, right? There are so many proteins and other molecules involved in any cellular process that it's almost impossible to completely reimagine that in a larger volume, essentially. And just getting the right enzyme in the right place to deal with the right molecule and have it and the energy transfer and things like that, these are immensely complicated processes that it's really not, I mean, maybe someday, but right now it's really not possible to kind of recapitulate that in the kind of the large scale. Yeah, go ahead.
I agree with you. I mean, my question is still, but like, how we we going to convince the customers or the people,
sure, yeah,
like about genetically modified? Do we need to do some human studies as well with that, or what? I don't know. Maybe I'm just
Yeah,
concerned
so,
about because when I buy food, I always see this is GMO, so I just have a little hesitant
yeah,
when I have to buy that. Just wanted to know like a basics of it would be daily
yeah,
life, how I can.
absolutely. So it's really GMOs have a very bad advertising problem, essentially, right? And scientists as a group didn't, I think, appreciate how much negative feedback there would be on genetically modified organisms, right? And GMO foods. So it's really a question of optics a lot of the time. I mean, we see it with vaccines as well, right? People don't trust that governments or scientists or Big Brother, whoever it is, right, has their best interests at heart. And so, we try to create educational resources to try to combat this a little bit. And we absolutely need to be kind of ahead of things when it comes to especially cellular agriculture, where it is going to be we're producing food and food ingredients. We need to get out in front of it and educate before they hit the shelves. Now, those non-GMO labels that you mentioned, right? For example, so in Canada, those non-GMO labels are completely voluntary and they're not regulated, which means they don't actually mean anything. And then when the label's not there, that doesn't mean there's a GMO inside, but human psychology, right, tells you, oh, that says non-GMO, and that doesn't say anything. It must be a GMO, right? And so there's a lot of kind of issues as far as marketing and education that, yeah, we can't fall into that same trap again. And we also, I was talking to a class, a food science class last week, and this question kind of came up there too. And they're asking, how do we convince people? And you know what? And you can provide studies, you can provide great information. At the end of the day, it's going to be kind of the flashiest message, the simplest message that gets through. And so there are lots of organizations who are putting those types of things together now, even though it's maybe a little bit too late. And then the other thing to remember is that there are always going to be people who just say, no, this is bad, and will not change their mind. Let's say that's 10% of the population, right? 60% of the population are totally fine with GMOs and vaccines and all this other stuff, right? And so it's the other 30% that we need to convince, and not, spend billions of dollars and all of our time on people who really are not going to change their mind no matter what you tell them. So it's difficult, it's tough, and there's no right answer. But I do think that through rigorous research, and not just the technical research, again, social research, looking at life cycle analyses, looking at consumer perceptions before we're producing these things, can go a long way to ensuring that they have that kind of wider adoption that GMOs are fighting against now.
Thank you, Dr. Mike.
Thanks,
Vikas.
All right. And I think we might have time for another question if you're okay there, Mike, for a little bit longer there, Mike.
Absolutely.
John, I see your hand up there. Welcome.
Thank you. Thank you very much for your nice presentation. I'm John at Inha University at South Korea.
Hey, John.
Hi, John. Yeah. You mentioned about the engineering biology, and you also suggest some several challenges to achieve the engineering biology. For example, modularity and decentralization.
Mm-hmm.
So, do you have any strategic planning to achieve these challenges, or any good examples ongoing in Canada, in any industrial sector or any communities?
Sure. So I think I'll start with the modularity, and I think that's kind of the easier one, right? And what often I think we find is, actually getting back to Peter's question in a little way, is we often choose an output and we choose an organism, and then we and then we try to fit as many inputs into that process as possible, right? Through little tweaks to the metabolic process of our organism of choice. And so there's lots of examples of that happening. I know in Toronto, for example, of things like that bio-nylon that I was talking about. And so we've it worked out with purified sugars quite well. We can convert that to the building blocks of textiles like nylon. But now can we get more kind of complex inputs like food waste or spent distillers grains or corn cobs or lignin from trees, right? Extract the carbon out of those and use that for the same process. So that's kind of where we're starting to build in modularity just a little bit, is in those inputs. A lot of that comes down to strain engineering and things like that, and testing different natural enzymes that, again, we know are going to give the same output, but might be able to take in different inputs. That's certainly happening here. The decentralization is tougher, and honestly, we don't have enough physical infrastructure, things like bioreactors and the more complex, downstream equipment that's needed to purify our outputs, right? To even be able to test at enough scale to know, that we can get it to work in place, let alone build something in that's a little bit more transportable, a little bit more, that we can just move it around as it's needed based on where that output's needed. So there's real there's technical issues there, but I'd say more than the technical issues are the financial and infrastructure barriers right now. And we're hopeful, and we're trying to contribute to that as well by building some piloting equipment and things like that. Yeah, that's kind of where we're at now. Is we're still figuring out the basics before we can kind of build in these things like modularity and decentralization. But yeah, it's being worked on for sure. Or maybe not as far as I'd like, but this is all very new still.
Okay, thank you very much.
Thanks, John.
Thank you.
And thanks for joining so early in the morning.
All right. And are there any other questions? I don't see any other hands up, but I think we have a couple more minutes if anyone would like to ask a last question or two. Okay.
Yeah, awesome.
Well, I think we'll wrap it up here for this evening then. And I just would like to mention to all the people joining us on the call today that we will be having more speakers throughout the spring. So please, if you're not already an IACCE member, please join on our website. Again, there's no cost for that. And you can be notified of our upcoming speakers and other events and such as we grow. So please join us for our next series of events as we come along. All right, well, thank you for everyone for joining us tonight. And a special thanks to our speaker, Mike, and for a very good talk. And we'll hopefully see everyone again soon.
Awesome.
Thanks.
Bye, everybody.
Have a good night.
Thanks again.
Thanks. Bye-bye.